It's always a pleasure to be with overcomers, and really, truly, I look forward to these presentations and chats. I'm going to go ahead and share my screen. Hopefully, you guys can see this. And sort of I made the title of my talk, Immunotherapy Breakthroughs for Ovarian Cancer, Hope or Hype. And spoiler break, I'm going to tell you that the correct answer is hope. And so with that, my fun fact is I recently completed a 10-day silent insight meditation or Vipassana retreat that was in sort of the ranch area between Austin and Houston. It was really beautiful. It was nice to be totally off the grid, unplugged, no phone, no computer. And the day started early at 4 a.m. This sunrise is not the beginning of the day. When we woke up, it was dark. But this is after the first sitting meditation from 4:30 to 6:30, where we were walking towards the dining hall and you could see the sunrise. And so that was definitely a wonderful experience that I just wanted to share with you guys as a fun fact. So what is immunotherapy? I know that Overcome has a diversity of a large audience, and among people on the call today, some people may be very familiar with immunotherapy, but others maybe not so much. And so immunotherapy is a cancer treatment that uses a patient's own immune system to recognize, attack, and kill cancer cells. And it's not one thing. There are many different types of immunotherapies. Many of you may be familiar with medications that fall into the category of immune checkpoint inhibitors. These are drugs like Pembro or Keytruda, Nivolumab, Opdivo, Atezo, Ipi. These drugs sort of sometimes we refer to them as taking the brakes off of the immune cells. So the immune cells can be inhibited by cancer cells because the cancer cells can sort of have shut-off signals on their surface. And so these drugs interfere with these shut-off signals. But there's also vaccines. There are cytokines, which are sort of growth factors for immune cells. And then there are cellular therapies where we actually transfer live immune cells and put them into patients. And these include types of immunotherapy called TIL or tumor-infiltrating lymphocytes, CAR immune cells, engineered T cells, NK cells, et cetera. And we'll talk a little bit about this. But for those of you, obviously in a 25-minute talk, I can't go into in depth about immunotherapy, but I wanted to share with you this resource from American Cancer Society. They have a nice immunotherapy page that has many different types of immunotherapies. And for those of you who are interested, you can always access this to learn more. And so some of you may have heard about immunotherapy, but you're wondering, well, why is there so much interest in this immunotherapy? What's the big deal? One of the reasons it's a big deal is that immunotherapy has provided benefit for patients with multiple cancer types. I think that drug Keytruda has 14 different cancer indications. And many of these cancers were cancers that previously had very few useful treatment options for patients, for example, melanoma. And the other interesting thing about responses to immunotherapy is that sometimes these responses can be very durable and continue even after drug discontinuation. And that's a little bit different than what results that we see for chemotherapy, for example. But there are important caveats as well, that current immunotherapies don't work in all cancers. And ovarian cancer, of course, is one that's near and dear to the hearts of everyone on this webinar. And that's one where we're still trying hard to make more useful immunotherapies work. And also, even in cancers where immunotherapies drugs are approved, not everybody derives benefit. In fact, less than half of patients might derive benefit. And these drugs can have autoimmune side effects that can be serious. And so there's a lot more work to be done to make better and safer drugs. So I'm going to share with you some of the bad news in ovarian cancer. So these PD-1 or PD-L1 immune checkpoint blockers, these are drugs like Keytruda, Opdivo, that inhibit with this PD-1 checkpoint. These drugs overall have shown shrinkage of tumor in only about 5 to 15% of patients. That's what we refer to as the response rate. And then stable disease are times where patients have a scan. The cancer hasn't shrunk, but it also hasn't grown. So some patients, the best response they get is about 20 to 45% of patients have stable disease as their best response. Unfortunately, even when there's benefit, the average time to progression is not great, so only 2 to 4 months. PD-L1 immunostaining can be a biomarker for successful immunotherapy in some cancer types. However, in ovarian cancer, it doesn't seem to really identify patients who might benefit or who might not benefit. So, when immunotherapies came along, the hope was that, if you compare the curve of conventional therapies, this is the curve shown in green here. So you compare the green curve to controls. So this is patients without treatment. The blue line, you see that this curve moves to the right, meaning that with conventional treatments, things like chemotherapy, more patients are surviving longer period of time, right? So that's what the curve moving to the right shows. But what you notice with immune checkpoint therapies is that maybe a smaller portion of patients benefit, but there's this flat tail to this curve. And what that means is a small portion of patients are deriving long-term benefit. So, the x-axis is time here. And so the idea and the hope was that by combining conventional treatments such as chemotherapy with immune checkpoint inhibitors, we get to the red dotted line, which is kind of this hypothetical curve of more patients living much longer. There have been several trials. I won't go through all of them, but I just want to give you an example of this trial where it was for patients that were newly diagnosed ovarian cancer. And the control arm here shown in the blue, it was patients got standard chemotherapy with carboplatin and Taxol plus bevacizumab or Avastin. In the experimental arm, patients got the same treatment, but they also got a PD-L1 inhibitor called atezolizumab or atezo. As you can see from these survival curves, the blue line, which is the sort of the control arm, and the red line are almost completely transposable. So really not much benefit. And they even looked at the PD-L1 positive population, and there was a slight benefit. But as you can see, the difference in average survival in the PD-L1 positive and negative, even in the PD-L1 positive groups, was only different by less than 2 months. So this was really largely a negative study. So immune checkpoint inhibitors by themselves don't work very well, plus chemo, they don't work so well. And so this has led some people, some of my colleagues to say, oh, well, immunotherapy doesn't work in ovarian cancer. Well, I disagree with that. And I think a more accurate statement is that we haven't figured out how to make immunotherapy work in ovarian cancer yet. And I want to emphasize the yet. But what I want to show you guys is what are some ways we're thinking about this. So how do you make immunotherapy work in ovarian cancer? And a big theme in our talks today is supposed to be hope and reason for hope. So I'm going to give you several reasons for hope. First is that we're currently using first generation of immunotherapies or immune modulating drugs. And there are advances in bioengineering, and I'm going to get to that a little bit later. But most of the data that I presented are drugs like PD-1 and CTLA-4. So these are drugs that Keytruda or Opdivo target. And you can see there are all these other targets, except that this slide was from a paper in 2018. So I only use this slide because it still fits on one slide. The number of targets now would probably cover 3 slides worth. And so the trick is to find out what are the key checkpoints that are most relevant in ovarian cancer and are present on ovarian cancer cells, and how do we find the right combination to make the immunotherapy work for patients with ovarian cancer. And I just want to give you an example. This is a paper that came out a couple of years ago in melanoma, where they, standard of care for melanoma was treatment with nivolumab, which is an anti-PD-1 drug. You see here in this illustration that PD-1 is one of the checkpoints that's blocked by nivolumab. In this study, they asked what would happen if you added a second checkpoint inhibitor, this drug called relatlimab, which binds to another checkpoint that's called LAG3, okay? And so what they found is that compared to nivolumab alone, which is the green curve, patients with relatlimab plus nivolumab had much higher median progression-free survival. So time to the cancer progressing was 10 months in the combination drug group versus only 4.6 months in the patients that had nivolumab. And this is an example of how combination immunotherapy can improve on results of single-agent immunotherapy. I do want to talk a little bit about advances in cellular therapies and bioengineering. So, really the science has advanced in this area. This diagram shows this figure, these things that look like letter Y are antibodies. And antibodies, you can think of them as tiny magnets that can go and bind to little iron beads, except their targets are not iron and they're not real magnets, but you can think of them as that way. And these magnets then can be designed so that one, you can put 2 magnets together. One end of the magnet can bind to T cells, which are important immune cells in our body, and the other end of the magnet can then target tumor-associated antigens, or TAA, as you can see in this figure. This is a little surface, think of it as a iron bead on the surface of the cancer cell. So these antibodies can go and bind to these cancer antigens. I think this morning you heard from Stephanie Blank about how these antibodies can now be tied to chemo molecules so that you can more precisely deliver chemo preferentially to the cancer cells, and not just chemotherapies that would be just administered intravenously can now be more targeting the cancer cells. Cellular immunotherapy is another great advancement. This is a living drug. What do we mean by living drug? That means we can take immune cells out of the body. We can increase them numerically in the lab. We can engineer them so that they recognize specific cancer surface proteins, so that when we put them back in the body, they can more effectively go and bind to and attack and kill cancer cells. And this idea is now FDA-approved treatment in patients with B-cell lymphomas and leukemias. So we know that this can work, and there's a lot of active research to investigate how to make this work in solid tumors. And so this has been really a great advance. These CAR T cells, as I mentioned, are FDA approved for some leukemias, but there's also significant toxicity to them, both clinical and financial. So these are potent therapies, but they can cause what's called cytokine release syndrome, which is high fevers, low blood pressure, multi-organ damage. There can be neurologic side effects. Patients may require ICU stays. And so these are definitely potentially significant side effects with these treatments. And, some of the costs, for treatment, maybe $500,000 to $1 million per patient. And so how can we do better than this? And one of the things we're interested in with some collaborators at MD Anderson is next generation CAR therapies, and here using engineered NK cells. And the NK cells are a different type of immune cells. We can use them from cord blood. So little babies that have cord blood donated, these cord bloods have a high number of NK cells, and they can be used to treat other people. So some types of immunotherapy, you can use other people's immune cells. For this kind of immunotherapy, it can be an off-the-shelf treatment where the NK cells can be engineered to recognize a number of different cancer-specific markers, and then they can be used. And each cord blood can produce enough NK cells for up to 100 doses. And so I just want to put in a plug for one of our studies that's using CAR-engineered NK cells that recognize a target on ovarian cancer and some other cancer cell types called TROP2. And this is a trial that's currently open. And the other nice thing about this treatment is we're actually putting the immune cells directly in the abdominal cavity, where of course ovarian cancer likes to spread and grow, and so and then I wanted to just show you these results are not for ovarian cancer. These are what we someday hope to get in ovarian cancer, but this is one of the patients of one of my collaborators, Dr. Rezvani, who published the first in human use of NK cells, NK CAR cells in lymphomas. And so these areas where you see the red circles are areas of enlarged lymph nodes. So lymphoma is a cancer of lymph nodes and lymph glands. And you can see that these black areas within these circles are areas of cancer. And you can see that 30 days after NK CAR infusion, you can see that all these areas have been cleared out. And so we're hoping to hopefully recapitulate some of these successes for patients with ovarian and other cancer types. Another reason to be excited is there seems to be, as I mentioned, the name of the game in ovarian cancer is going to be what's the right combination. We know checkpoints alone are not useful, but there's some data on drugs that are called tyrosine kinase inhibitors or target a growth factor called VEGF, vascular endothelial growth factor. VEGF is also targeted by Avastin, and many of you in the audience may be familiar with Avastin as a drug that's already used in ovarian cancer. And not very long ago, endometrial cancer was like ovarian cancer, was a cancer that didn't have a lot of immunotherapy work for it. This was results of Keytruda by itself in patients with endometrial cancer, and only about 10% response rate. But when Keytruda or Pembro was combined with one of these tyrosine kinase inhibitors called lenvatinib or Lenvima, that increased the response rate to 36%. So from 10% to 36%, and that happened in endometrial cancer. But in fact, there was results a few years ago in ovarian cancer where similarly patients that were treated with combination of Keytruda and Lenvima had a response rate of about a third of them had responses, so about 32% response rate, and another 40% of patients had stable disease that was not growing but also not shrinking. And then last but not least, I want to share with you this study from Roswell Park. This was exciting because the investigators used Avastin, Keytruda, and an oral chemo drug called Cytoxan or cyclophosphamide. So they used this 3-drug regimen, and what they found was in all comers, a response rate of about 47%, but even in patients that had platinum-resistant disease, there was a response rate of 43%. Again, remember, drugs like Keytruda by themselves have a response rate of only 5 to 10%. So this is significantly better. And these results, in fact, were so strong that even though this was not a randomized study, this was just a single-arm study, I should also mention they found that those patients that had less than 3 prior lines of treatment had longer benefit on average than compared to those that had more than 3 lines of treatment. But this regimen just recently became listed in NCCN guidelines. You heard Josh talk about NCCN guidelines, and just like there are guidelines about genetic testing, there's also guidelines about treatment of cancers, and sometimes drugs that are useful but not FDA approved, if they're listed in the guidelines, we can prescribe them, and insurance companies often will cover the cost if they're listed in NCCN. And so I wanted to highlight this treatment because it's the only, quote, standard immunotherapy options for patients with ovarian cancer. There are no FDA-approved immunotherapies for ovarian cancer, but this is the only standard immunotherapy option. And then last but not least, I just want to tell you guys a little bit about something that we are interested in, and that is to test drugs earlier in the journey, in the cancer journey of patients with ovarian cancer during the phase that is referred to as minimal residual disease. And so this is a part of a breakthrough cancer funded project where it was, we received $15 million for 3 years, and this is bringing together 5 large cancer centers listed here on this slide. And the idea is that if we think about the number of cancer cells in the body over time, you might imagine that number increasing until eventually a patient is diagnosed. With frontline treatments, surgery, chemotherapy, that number then goes down until it drops below this dotted orange line. This dotted orange line is meant to represent the limit of detection of CT scans, CA-125, sort of clinical detection. And for many patients, become sort of NED or undetectable or in clinical remission, only to find that over time, unfortunately, the cancer becomes detectable again. Then we treat patients with additional lines of therapy, and then only then we sort of think about trials of new treatments and clinical trials and investigational therapies. So the sort of the idea here is that what if we looked with a second surgery after frontline treatment to see who might have residual disease that's surgically detectable, even if it's not apparent on CT or by CA-125. And then for those patients that we detect disease, we can introduce investigational treatments, targeted therapies at that point, rather than wait till the cancer takes hold again. And then this rate of minimal residual disease can also accelerate frontline treatments because it can tell us right after somebody's finished frontline treatment how successful that frontline treatment might be, so that clinical trials for frontline treatments can be, we can have more trials testing new approaches since the current drugs that we're using, we've been using for about 40 to 50 years. I just wanted to sort of say, how are we kind of making sense out of this? Our first trial for patients that have minimal residual disease was just with Avastin. But unlike maintenance Avastin, these patients receive Avastin until the time of progression. On average, that was about 8 months. And now, remember, the same drug regimen that I told you about, that's currently the only immunotherapy that's available for patients with ovarian cancer, we're introducing this regimen at the time of sort of minimal residual disease. So our follow-up study, our follow-up clinical trial, again, looks at patients that have surgery documented residual disease who are not candidates for PARP inhibitors. And so these patients can enroll on this trial. And our hope is that we can extend average time to cancer progression by at least 6 months. So that's sort of the hypothesis of the study. And then we have another trial where it's for newly diagnosed patients that are going to get neoadjuvant treatment. I just want to point out this trial. It's basically standard neoadjuvant chemotherapy versus standard neoadjuvant chemotherapy plus an intraperitoneal immunotherapy called IL-12, a drug called IMNN-001. But the interesting thing about the study is that it's the first study to actually use the rate of minimal residual disease after frontline treatment as the primary endpoint. So we're going to see how many patients in the experimental arm had minimal residual disease and compare that to how many patients in the control arm had minimal residual disease. And so that is going to hopefully accelerate how fast we can identify useful drugs in this frontline setting. So in summary, I hope I've shown that just adding chemo to immunotherapy in ovarian cancer hasn't worked. So the kitchen sink approach hasn't worked. There tends to be synergy between tyrosine kinase inhibitors or VEGF inhibiting drugs and immune checkpoint inhibitors. This is promising and deserves additional investigation. Biological engineering can produce more potent immunotherapies and cellular therapies that can be living therapies that cells that divide inside the body and hopefully can work towards controlling the cancer. And understanding the minimal residual disease phase of ovarian cancer may provide novel insights for rational therapies at a time when there's the fewest number of cancer cells in the body. And so with that, I just want to thank all the overcomers for everything that you do, and especially Runcie and the Overcome team for putting awesome programs like the Zeal for Teal. Thank you all.